Good evening, everyone. I'm so glad that you could join us tonight. It means a great deal to have this community together as we move closer to treatments and one day a cure for CMT. For those of you that don't know me personally, I'm the newest member of the CMTRF community, and I've been CEO for the past 5 months. Although I'm new to CMTRF, I am not new to CMT. I spent all of my life as an athlete, running marathons, biking century rides, and staying very active. So when I began to lose my balance and coordination, it was really scary. It was after meeting CMTRF and this community that I finally had the courage to get my genetic test. I learned that I am joining my paternal grandmother, three uncles, and my father as a CMT2W patient. So, like many of you, I know firsthand the weight this disease carries and the urgency that we feel every day. And that's why tonight is so exciting. We're here to celebrate a major milestone. Augustine Therapeutics raised $85 million in Series A funding to bring their lead candidate for CMT into clinical trials. I'm going to say that one again. Augustine Therapeutics just raised $85 million to bring their lead candidate for CMT into clinical trials. This is the kind of progress that we dream about, and it's the direct result of strategic investments, relentless science, and of course, donor support. The CMT Research Foundation was one of Augustine's earliest investors. Your donations helped put them on the map. That early funding gave Augustine credibility and momentum. That is exactly how your support gets multiplied. We take every dollar that you give us to open doors, accelerate science, and draw in larger funding partners. So now it's my pleasure to get the call started by introducing CMTRF's co-founder and Chief Mission Officer, Susan Rudiger. Susan, take it away.
Thanks, Laura. It's so great to have you here. And thank you to all the participants who are here tonight. Again, it's such an exciting evening for us to celebrate another massive investment in CMT research and drug development. So thank you. I look forward to introducing Gerhard Koenig, the CEO, and Andy Hugh, the Chief Business Officer of Augustine Therapeutics, to tell you about their project and what their plans are for using this $84 million to bring it into the clinic. But first, we're going to hear from Ryan Aguskiza, who is after we hear from Gerhard and Andy, we're going to hear from Ryan Aguskiza. She's our Director of Research, and she'll give you her perspective and the CMT Research Foundation's perspective as patients on how we feel about this project. But I know you guys are going to have questions along the way, so please, down at the bottom of your screen, you'll see a question and answer button. Go ahead and write your questions in there as they come up. We'll be fielding them throughout the entire session. And at the end, we'll open it up for a Q&A. I'll read some of the questions and hope to try and get to every single one of them. But if we can't, we'll do our best to answer them just to you in writing. So without further ado, I'd love to welcome Gerhard Koenig and Andy Hugh from Augustine Therapeutics. Thank you so much for being here.
Thank you
for having us.
Thank you. So good evening, everybody. So my name is Gerhard Koenig. By now, or maybe over the next few minutes, you will realize that I'm not originally from the United States. I talk to you from Massachusetts. I work from here, from Newton, which is a suburb of Boston. But I was originally born in southern Germany. Got my education there, got a Ph.D. there at the University of Heidelberg, which is a cute little town in southern Germany, very cute university and very good. And I was trained as a scientist. Telling you a little bit more about myself so that you know who I am and who we are and why we are in this. So after my PhD, I did a short postdoc, and then I decided I wanted to go to industry and ended up at Bayer Pharmaceuticals, or Bayer Pharmaceuticals, as we used to call it. And I spent 10 years there, working mostly in discovery. And 5 years of that was, again, in the United States, in Connecticut. They used to have a research center there. And 5 years was back in Germany. I met my wife actually in Connecticut, so I have a true American family now. Two of my kids were born actually in Germany, and one of them, the last one, was born here in the United States. So she's our real American of the kids. So I love it here. I like working from here. And after I, was quite satisfied with my work at Bayer, I worked mostly in neuroscience. Bayer made the strategic decision to really get out of neuroscience. And I had a choice to make. And as you know, sometimes you get calls, and I got a call that maybe there's an opportunity for me back in Boston, actually. And that really, Boston was calling, and I got back to Boston to become the CSO of a company which was later known as Forum Pharmaceuticals. And again, that was in neuroscience. So you see a pattern here. I'm really passionate about the science, and I like the neuroscience in specific because that's what I was trained in. And there I was for 14 years. I was the CSO of Forum Pharmaceuticals, and we had a range of different programs all the way up to phase 3. Unfortunately, that phase 3 was not really repeating itself in its efficacy, what we have originally seen in phase 2. But we had a range of other programs, but the company decided to close down. And then I joined afterwards as the first-time CEO, a small company which was focused in pain. And the idea was really, and that was in 2016, that was funded out of Atlas Venture here in Boston, in Cambridge, focused on chronic pain, but non-opioid. And you all know this is a big problem. Opioids really are dominating, of course, and they're useful, but they also have, some liabilities coming with it. So the goal was really to have non-opioid treatment for pain. Unfortunately, we had some toxicity early on in the animal studies, and so we closed the company. That's my next station was then actually to fund my own company, which was Arcuda, for which I was the CEO for about 5-6 years. That's actually where I met Andy. Yeah, I hired him. He was early on our chief business officer. And Arcuda really was sold to Johnson & Johnson. That deal was really finalized in January 25. You might have heard about it or not. But it was really put on its way early on in 23 and then in 24. And that's relevant for the history when I tell you a little bit how Augustine really came all about. So keep that in mind, but I will revisit that a little bit, the timelines. So maybe I'll let Andy introduce himself and tell a little bit about himself as well and why he works with me.
Thanks, Gerhard. So again, thank you to the CMT Research Foundation for having us tonight. It's really exciting to be able to share the news, obviously, and then more of the story about Augustine. So just quickly on my journey, I'm actually originally a physician by training. So I started my career actually trained in pediatrics, and for a time was considering going into pediatric neurology. But my career took a bit of a left turn, and I've spent the past 2+ decades working on the business side of things in the biotech industry. And I've been in a variety of capacities. So I've seen a lot of market upturns and downturns. I've been an investment banker, strategic consultant, and then more recently for the past 17 years, have been with companies, large and small. And for the past decade, I've had the privilege and pleasure of working at emerging companies focused on rare disease. So events like tonight are my favorite thing to do, and we don't get to do it often enough, but the opportunity to interface with and interact with patients really feels like full circle to me, going back to my early days as a clinician. Certainly something I miss, and certainly something I really relish the opportunity to just certainly share our excitement with. And so, yeah, Gerhard and I, so I worked at a gene therapy company before Gerhard and I met up. The programs we're working at there are actually now in phase 3. So it's very exciting that those programs have lived and continue to flourish. Gerhard and I connected at Arcuda Therapeutics. We're also focused on rare disease, in that case, an inherited form of dementia. And then after that got sold to J&J, Gerhard came a knocking, and for better or for worse, I picked up the call, and here we are again. And we couldn't be more excited to share with you the really, really unique opportunity we have in front of us, and certainly that's been fueled by the support of the Research Foundation. So I will, I'll go ahead and turn on the slides here, and we will walk you through a few slides to share a little bit more of the story and our excitement about what's to come. So.
Right. So now I gave you a longer introduction about myself, and I emphasized a few key events on a timeline. And so you will see that again here when we talk about Augustine. So it really started all with really groundbreaking discoveries by this gentleman here in his lab on the way on the left on your screen, Ludo van den Bosch. It's a Belgium Flemish, so they speak Dutch there. Belgium has two different languages, actually three, Flemish, which is Dutch, then French, and a minority of people also speak German there. So it's a very diverse country with respect to cultural input. So Ludo actually in Leuven, which is in the Flemish part of Belgium, has a big lab, is very well known, and has already in 2011, as you can see in Nature Medicine, which is extremely prestigious, is a very prestigious journal, published findings that HDAC6 has potential when we inhibit it for diseases such as CMT, but he later on published also other key findings, not just CMT1 and 2, but also, for example, other things which cause neuropathies. And some of you might know, for example, when you take chemotherapy, when you have to unfortunately undergo chemotherapy, that can also damage the nerves, and that comes down to people have then suddenly, these sensational problems and have neuropathies similar but not identical to what you unfortunately have in CMT. So Ludo was really the one who had published this. And then if you click once, you see a group of investors, which are mostly local. PMV is a Belgium institutional fund. VIB is the Institute for the Flanders Institute for Biotechnology. That's sort of a tech transfer unit. Then you had VBio, True Venture Capital, and AdBio is a French small investors. They came in and really provided the seed for those findings, and they wanted to move it into from the academic setting into a more industrial. They hired this gentleman here. Many of you might know him, Sylvain Selenier, as the CEO. They gave it the name Augustine. And Sylvain really had the task and did an excellent job to really, with this minimal funding, to get going, industrialize the effort. That means hire a chemist, hire some people who work at Augustine. It was a very small team, and moved it forward in the chain. And then I was really approached. If you click one, he also did the FLYO grant. That's a Belgium special grant, which actually helped the company to really get going with the early discovery work. The compound we really pushing forward right now and very soon start the phase 1 has the number, it's a long number, HET-100216. We call it 216. That is the discovery which was made early days in the labs in Leuven, even when it was not fully spun out. So that was the discovery, and Sylvain and a few scientists who worked with him really pushed that forward. I was approached in 21 and then really joined as chair of the board in 22. And one of the reasons why I joined, because I always have followed the field on HDAC6, and I've found it an intriguing, including some of the science I just mentioned, intriguing target for a number of indications, including CMT. What was missing is really good compounds, compounds which can be used for a chronic treatment. There are HDAC6 inhibitors, but they're not really the best suitable compounds in terms of how they behave in a body. They have side effects, and they are okay for things like oncology, but for a chronic treatment, I wouldn't recommend it. And so that was missing. When I was able to look deeper into the science, I realized that the real true jewel of what Augustine has come up with is the discovery of novel chemical entities, which have the potential to really go all the way. CMT, we mentioned that before, of course, really came in very importantly, not just the money, but also the recognition that the foundation really supported the work here. That was really great to see that coming in, and it helped us. Sylvain and I very quickly hired then Frederick Romboots. He became our head of discovery. He's an excellent drug hunter. He's a medicinal chemist, and he expanded the pipeline in the small team, meaning that we have now more compounds, not just 216. That's important because you want to have backups, you want to have second generation compounds. It's important from an overall value perspective to have that. If you click one more time, you can also see that at that time we have actually received also the foundation support from ALS, which is, as you know, a very devastating, fast progressing disease. The difference there is that the potential was shown in academic labs that there might be also HDAC utility for ALS, but you need these compounds to get behind the blood-brain barrier, meaning those compounds have to have a different properties. So 216 would not be good for that. So with that additional help, we were able to also start those programs. So this is a different program than 216. Now it comes clear that in 24, when I had a little bit more bandwidth, I was approached by Asabis, who is part of the Series A, the big Series A, and Lilly, whether I would be interested to step up to really work with Sylvain and really raise more money because we're now getting into the clinical trials. That means we need really the fuel, the money to do all that and go all the way, hopefully, without immediately depending on other input again. And I was able to do that. And then, you know, the most work was done in 24 when then Asabis, Lilly, we hired the CMO. I forgot him, Carl. He's instrumental, and I hope you will have an opportunity to have him present to you at some point as well. He's a neurologist like Andy. He has done many clinical trials, very, very good scientist as well. I became the CEO, and those names here are important: Novo Holdings and Jato. These are the key investors who came in with a lot of money for the total of, as you see here down, 85 million. They really led the second part of the round. And all together with Lilly, Asabis, Novo, Jato, and Newton Biocapital, we were able to raise that 85 million or so. All the work was done for the fundraising, I should emphasize, really in 24. We announced it in early 25. I became officially the CEO, as you know, in January. And, you know, I'm now, like 4 months into the CEO, so I'm learning still a lot about CMT, and I'm excited talking to you. But the key message is here, we can now really go into phase 1, and we have the money to also do the subsequent phase 2, which is listed here. So for timeline in 25, in a few weeks from now, we hope to be able to start the phase 1. Now that will be only in healthy volunteers, and also we have a cohort which is a little bit older. So this will not be patients yet. And then if that all goes well, and we can certainly talk about what goes well means later on in 26, in the first half of 26, we plan to start, if it goes according to plan, to start the phase 2, which is an efficacy trial in patients. So that is a lot of data, a lot of things, but this is how it all came together to be in the right moment at the right company. And also, you know, I think Andy should probably comment on this. When this all happened in the second half of 24, when we got the term sheet, I think we cannot overemphasize how, I wouldn't say lucky, but how fortunate we are. How fortunate we are that we have raised so much money. It hasn't escaped to you that right now there's a lot of uncertainty in the market, investors getting concerned about the future, about the predictability of the economics. And to have been able to actually raise that amount of money allows us to not be immediately exposed to the financial turmoil out there. So our goal right now is to do good science, to get into human trials, and really advance this program as much as we can. So Andy, I think you have a better perspective about this than I.
Sure. Yeah. I mean, I can provide a little more color just in terms of how wonderful this news is, really. And I think part of it is taking into consideration also the context of the industry in which we work and the broader global context. So obviously, even thinking back to kind of the early days of the company's formation and the early progress and the early investment, I mean, that was occurring during the pandemic, right? So it's we went through a similar thing at Arcuda, which was to find a way to continue making progress even within the confines of the pandemic, figuring out how to get people back in labs safely, figuring out how to continue the mission. Because like Augustine, we were focused on rare disease patients. And so we the clock's running, and we know that we want to every day, every hour matters. And so for the biotech industry, it was also interesting because coming out of the pandemic, there was this period of sort of a lot of excitement around investing. And so there was a brief window where capital really flowed fairly freely into companies. But then we started getting into the 22 and 23 timeframe, and it started getting harder and harder for companies to raise money. And it was a broader macroeconomic situation. And so for a time, I remember, the beginning of 24, every year we go to this meeting in California called JP Morgan Conference. And, coming out of 23, the meeting is like, oh, there's optimism, kind of in the air this year. And I think 24 played out, and that optimism was short-lived, and we continue to kind of be in a situation where it is really, really challenging for companies to raise capital. So when Gerhard talks about the fundraising coming together at the end of 24, it's a testament to the science for sure. It's a testament to the validation provided by CMT Research Foundation's involvement, and it's also a testament to the unmet need here, and the realization from investors that this is something that where patients really need the help. The amount of the funding is of note too. Back in the day when I was helping companies go public, 85 million was sort of what we call like a standard IPO number, and that would be a number that companies would go public on. In this case, we're staying privately held. And how far that gets us in terms of clinical development is also super important, right? Because as Gerhard will go through, this is meaningful in terms of it's enough capital to actually get the lead program through a really meaningful information point, and really to the precipice of being, in a phase 3 trial to ultimately help patients someday. So for context, Garrett and I have lived through a lot of these up and down cycles. This is a unicorn situation in terms of the investors that have come in, the amount that's been raised, and our ability to really deliver what we hope some really promising new insights as we move our pipeline forward into clinical trials. So I'll hand it back over to Gerhard.
Yeah, and just to add a little bit more to that, it is right now the largest round Series A in Europe this year, and it has the chance to be the largest round, for a European company. We have American investors, obviously, but for a European company, it could be all of 25, right? There is a chance that this will be the largest round. That's at least what other Europeans tell me. So we got a lot of attention from the Europeans. I would also say that maybe it is just luck that, me being originally from Europe, I probably have understood the localities a little bit more. I know the university, I know of the quality of the science, what they're doing. That maybe is also helped us here. Europe is not quite as hunted, so to speak, in terms of tech transfer opportunities. So that helped us here too. So, maybe there's a little bit luck in there too, but we're excited. What drives me, just that you know who I am also personally, I'm very direct, I'm very candid, I get passionate about the science, and when I see something, I get very convicted about it and have conviction going forward. And I want to show you one slide where I probably know myself, not this one yet, but we'll get there. When I see the data, I get really excited. And so I'll want to hopefully can share that with you, why I think this is really very promising. So we talked a little bit about Ludo. The one thing I also should say, I've worked over my years with many academic founders, and all of them are smart, all of them are highly published, but also a lot of them have a big ego. Ludo does not have a big ego. You go out with him, you talk to him, he brings other academics in, and they say like, you should talk to this person, you should talk to this person. He's a true connector with other scientists, and I like that. He was extremely instrumentative in getting this going. So here you see again some of the publications, and they're listed also to tell you a little bit the diversity of applicability here of HDAC6. Obviously, here Charcot-Marie-Tooth, but also for different causes, different genetic inducers here, in animal models, which cause Charcot-Marie-Tooth-like symptoms. And the last one here on the bottom here is TDP-43. That is obviously a CNS disease that has to do with ALS. But this is a pathology also which can be found in Alzheimer's, for example, in amyloid plaques. So now you might hear a little bit, oh, that's why maybe we're also potentially interested in other applications for our pipeline. We are committed to CMT, and CMT is what really drives the value right now because we're going into the clinic with this compound, and that means it's more de-risked. This is a compound, we know its behavior, and now we have to actually do the human trial. The other aspects of the portfolio are earlier, and therefore there's more risk still baked in. So coming back to Ludo, Ludo drove this. He connects us with there. I have just the other day, I asked him a question about a quote. I get an answer within minutes and no fuss, so to speak. As I guess as a new American, I would say, sometimes they want to immediately know what's going on. He knows that I will tell him when we have something to say. So I really want to highlight that having the right founders really is important. And a small company is a team, and teamwork is extremely important, and you want to have fun. Research is frustrating and hard enough, and you need to really be asking the right questions. You need to be critical to each other. But as a team builder, which I see myself and somebody who likes building a new company, which I've done a few times, it is really important to have the right people, that they really work dedicated. And Ludo is one of those. So I think I said enough about him, so that's that you know that. So now let's talk science a little bit. You all know this, that we can classify CMT in the secondary and the primary. And what I show you here is, if the video goes, you see those, these are cells, and what you see here are axons. These are basically, or microtubules, sorry, these little lines which look like a net, like a spider web maybe. These are microtubules. These are the highways of a cell. And what you see moving here, these little kind of white dots moving along, are basically cargo which moves in the cell, which is this white glowing white blob on the left up here. And you see, this is not a neuron, this is a cell, but it's fundamentally the same in all cells. When you have those highways, those microtubules, which in the cartoon on the left here is a neuron. As you know, neurons can be very long, up to a meter or longer, a yard, several feet long. And those highways are extremely important to get the cargo transported from the cell body all the way to the end of the neuron. And that is also why neurons are so sensitive to any kind of insult, whether it is genetic mutations, whether it is toxins, we mentioned chemotherapy, whether it is such thing as aging is also, a risk factor as you get older, which then of course, means integrated exposure to toxins as well, sunlight and what have you. Neurons are the ones who usually suffer the first on transport issues. So when you have a problem with this highway, with these highways, and the cargo cannot be transported, and the cargo, for example, is mitochondria. Mitochondria are the nuclear power plants of each and every cell. They're very important. They make the energy equivalent so that the cell can basically fire up whatever they need to do. Mitochondria get transported along those axons, along those microtubules, this inner line here. Endosomes and lysosomes, these are cargo vesicles which contain certain nutrients, or they can also contain certain neurotransmitters. This is what the neurons are very sensitive. If those things don't get transported, they might suffer. They also go both ways. They go down and up in this case, in the cartoon here on the left-hand side. So the reason why I introduce you that is we have evidence, the field has evidence that HDAC6 and HDAC6 inhibition, it's an enzyme, affects how the cell can transport those vesicles, those cargo vesicles. And that is how we believe our efficacy, which we see in the animal models, which I'll show you in a minute, is really mediated by modulating how the cell can transport more effectively down to the axon certain nutrients or certain cargo vesicles or the mitochondria. And it again goes both ways, also taking away certain things they need to from the end of the axons. So HDAC6 plays a role in the transport. That's established. We will work more on this mechanistically, but in the meantime, we move forward with our compound because we have very exciting data, which I'll show you in the next slide, which is complex, so we go slow. So this is what I call the money slide. What I mean by this is when I saw those data, where I said, like, I want to work on this. And I could predict when I showed this to Carl, our CMO, Chief Medical Officer, I knew when he will see those data that he will be sold and wants to work with us. So let's go through this now. So what you see here is a mouse, in this case, the CMT1 mouse. And you see that this mouse, after 8 weeks, is because it overexpresses that transgene. After 8 weeks, it starts developing a certain phenotype. In this case, what we measure here is the amplitude. This is electric activity in the neurons in that mouse. You can see red compared to black is about 60% of normal. Black is 100%. And red means they already have some deficit in the electric activity. And then we start at that 8-week time point dosing in low, medium, middle, high, and high. So from low green to high green. And we started at 8 weeks. Now, when you dose 1 or 2 days, you see nothing. But if you dose for 4 weeks, from 8 to 12 weeks, that's the second part of the graph, you see that the low dose doesn't do much or doesn't do anything. The mid dose is actually reverting that. It goes back to the baseline where they started out at 8 weeks. And then you see even the higher doses can move it up further. Not all the way, but they can definitely revert some of the phenotype. So that's number one, evidence number one. Now evidence number two. This is now where we measure the speed by which the signal translates down to the nerve. Again, in the same mouse. Same picture. 8 weeks for a few day dosing, nothing happens. As you go for 4 weeks, you can see that actually it's almost mimics what you see in the left-hand side. You see that we increase the speed, the velocity by which the signal transports. So this is exciting. That one, you now see something more relatable. This is now not a biomarker or something. This is actually true what in this case, mice can grab with their paws. It's basically a measurement of their strength. And you can see also that they can go back almost to 75, 80% of normal in the mouse after 4 weeks of treatment, especially for the higher doses. So I want to stop there for a moment. I have a few gray hairs. I've seen a lot of failures. And I know that certain things are great, but sometimes don't reproduce. And that's important for you to hear. So this is exciting, but this is by itself didn't push me over the edge yet. What really pushes me over the edge is now this one, in a positive sense, I mean, is this down here, because these are translatable endpoints. So what do I mean by that? The two S's I'm showing you here in the bottom are things we can actually do in a very similar way in humans. Up there, we cannot do electrophysiology so well, it's more heterogeneous. But what we can do is in plasma or in blood, we can measure the acetylation of alpha-tubulin. That is, alpha-tubulin is part of the microtubules, the highway, which I told you. So this is basically the highway. And the microtubules are constituent of that highway. That's what HDAC does. It affects how tubulin is modified, and that affects in terms how good the highway is transporting the cargo. If you remember all the important cargo vesicles which need to go up and down. And what we can see is with the same dose response, meaning more, in this case, we see that the acetylation levels in blood cells is also altered. You can see that here. That is something which we will do in phase 1 in healthy volunteers. We will dose the humans, first a single dose and then multiple dose, and we measure this in humans. And the reason why this is so important, because this has been done before with other compounds in humans, which also showed it in animals, but those compounds didn't progress because the chemistry was not as good. Our chemistry is unique. We have the secret sauce, which we never show anybody. Even if we talk to investors, they have to sign 5 documents before they see anything, and then we don't give it to them because it needs to be protected. It's our intellectual property. So this is the secret sauce of Augustine, and this is what the team did early on. The second point is here, this one here, this is the neurofilament light chain. This is something which normally should not be in plasma. When it's there, it means that neurons released it from the intracellular content into the outside. That is in general a sign of neurodegeneration. NFL is up in ALS, in Alzheimer's, in FTD, in all kinds of diseases, and it's also up to some extent in CMT, not as strong. But the point here is when it's up and we dose, we get it down. This endpoint, by the way, was leading to the approval of an ALS drug without yet formal efficacy. So this is established now as a true surrogate. It's the first time that a drug was approved in ALS with a surrogate. So I mentioned that because this is the money slide. This is what investors, when I tell them that and I get excited, this is what they pick up. This is the data which made me certainly excited about the potential of those compounds. Carl, as well, as I told you, and everybody else who works with me will hear that from me all the time. So with that, and we can certainly talk more. I just want to make sure you also understand that we're building a team. So Andy officially is not yet the Chief Business Officer, but I want him to be the Chief Business Officer, and he will be the Chief Business Officer because he knows I am relentless. He will come. Then Virginie Kataj is our new Chief Financial Officer. She will physically join next week in Leuven, has already been part of the team for a week now. I will see her again in 2 weeks when I'm there for a whole week. I'm very happy to tell you also that we have soon a Chief Scientific Officer, and I met her yesterday, and she will sign, and she knows that I will not give up. She will come. I told her about the data, and she's very excited to join us too. So that will take another few more weeks. We talked about Carl. I'm very happy to have him on board. He will play a very important role for our clinical trials, of course. Then Sylvain, I don't want to forget to mention him. Sylvain was the CEO. He's now the SVP of R&D operations. He has been very instrumental in the background. He's very loyal to the company, gets a lot of credit for really the company getting to a point where I stepped in, and he's doing well. He's doing really well, works with everybody. He's a Swiss knife. What I mean by that, he can do lots of different things, and he's very engaged, and that's great to see. Frederick, I mentioned him, and Dirk. Frederick is our drug hunter. He's the managing all the optimization of the compounds. And with that, I'll stop because I know I have a certain time. But as you see, once I talk science, I get really carried away because it's great science.
Thank you, Gerhard and Andy, for that great update and information on the project. So before I give my comments, I just like to give a bit of a background for those who may not be familiar with the CMTRF and what we do. Our mission is to identify and support projects and technologies that advance the development of treatments and potential cures for CMT. So we provide funding and other resources to really help accelerate this work with the ultimate goal of just bringing effective treatments to patients faster. And, since 2018, we have funded 25 research projects, which have so far led to the development of 5 preclinical drug candidates. And one of those is, the one we just heard about from Augustine. And so we're very, very excited about that and to hear about this progress. And congratulations again on this amazing, impressive fundraising round. And so to go back to the project, our collaboration with Augustine started around 2 years ago when we funded a project to help optimize their novel HDAC6 inhibitor and test it in preclinical models of CMT1A. So we invested in this project because we knew it was, one of those unicorn projects, as Gerhard said. We saw the strong scientific rationale for HDAC6 as a potential therapeutic target, not just for CMT1A, which is the most prevalent form of CMT, but for other subtypes as well. And as you could see from Gerhard's presentation and Andy's comments, there was the potential to expand even beyond CMT, which really helped move that program fast. And additionally, we had just great confidence in the Augustine team's ability to execute and advance this program. So seeing them successfully complete this project and achieve this extremely impressive fundraising round really just validates our early support. And we are thrilled to have played a role in getting the project to this stage. and to help generate that data in that money slide. That was, you know, CMTRF support right there. And we're especially proud of just how quickly Augustine has been able to progress this project, and so excited to hear that clinical trials are going to be starting very, very soon. So big thank you to Augustine team for just being outstanding partners in this project and for staying committed to your mission for developing a treatment for CMT. We really look forward to the next updates and milestones ahead, and congratulations again. So I think now we'll open the floor to the Q&A, which will be led by Susan.
Hey everyone. All right, Gerhard and Andy, that was fantastic. Ryan, thanks for your perspective on it. So we have been getting a bunch of questions in, but if you all have more questions, please put them in the Q&A. But the first one, Andy and Gerhard, is for you. And in 2011, when you showed Ludo's first work, it was for mutations with HSPB, which is heat shock protein beta 1, I think, and vernacularly known in the CMT community as 2F. And then on your data slide, you showed that it was for 1A. So can you talk a little bit about the role that you think that your drug will take in the different types of CMT, maybe specifically, maybe a little bit more generally, and then maybe comment on your clinical trial plan for we know you have to do the healthy volunteers first, which is phase 1, but once you do get past that and get into patients, what is your clinical plan? Are you recruiting 1A more? Like, what does that look like for you? And we've got a lot of people on the call with not 1A, and we were getting so many questions. What about my type? And
Yes.
so can you talk about that journey a little bit?
Yes, absolutely. This is a very good question. And the good news is I anticipated the question. So because it's a good question. Let me just explain a little bit what we think, how we think about that. So to make it the answer quick, the phase 2 right now is planned for all comers. So we will not differentiate it according to genotype. That's the most important one. Now, why do we think that way? Why is Carl thinking that way? Why are we thinking that way? I did talk a little bit about this when I talked about the neuron is long. Remember that? It's not just genetically what can lead to actually the death of a neuron, you know, as it grows back, basically as it retreats itself back to the soma. There's multiple reasons why that can happen. We know from literature data from other groups, but also our own data, that we have several different genotypes which can be rescued with HDAC6 inhibitors. So that's number one. What we don't have is all 80 or 100 mutations, right? So just to be clear, we don't have that. But we have several of them. Some of them are primary and some of the secondary, which I showed, right? So some of them are more starting with a myelin deficiency, the other one more what's classical axonal deficiency. But also remember what I said, and I didn't show that because it's not about chemotherapy-induced polyneuropathy or neuropathy here, CIPN. We have beautiful data that it also works in this, where you just take taxanes, you know, chemotherapy, let's say cisplatin or those kind of agents, they can cause also a very strong phenotype in humans and in animals. And we have shown in animals that we can reverse those phenotypes by grip strength, by pain sensitivity, electrophysiology, and of course by NFL. And so that's not relevant for the audience, but it's important that there are different ways as scientists, when we look at this, to say, in general, when neurons get damaged, the potential for HDAC6 inhibitor is there, not just for one or two genotypes. So now I'm being a little bit too scientifically precise with my final comment. Could there be a responder which maybe responds better than others? Scientifically, that is absolutely conceivable. That sounds like a legal term, what I'm using now. That is conceivable. And we will continue working on this, right? So might there be some genotypes which respond a little bit better? That could be, but we don't have the data. Nobody knows that right now. But what I don't want to do is to wait with that and say, let's be very, very precise, and one or two genotypes might not respond as well. I don't think that is the right way to do this. And there's two reasons. It's not the right way, I think, if I were to be a patient, right? Which I'm not. but it's for the patient to decide. But it's also from a value perspective. I want to demonstrate to the world and to you that this treatment has potential in CMT. And we will analyze later on, that's just good practice, whether there are responders which respond better or not. But that's a long answer, so it will work in other genotypes for sure because we've tested it. Sorry, it took a little bit longer, but I wanted to get this across.
And just for clarity, for people on the phone who don't know this, it took me a little while to learn it. A genotype means what your genetic mutation is.
Correct.
So what he's saying is for multiple genotypes, it means for multiple types of CMT. And then when he
Correct.
talks about phenotypes, he's talking about symptoms. So
Correct.
that whether your symptoms start with a deterioration of your axon or your myelin, it sounds to me like, Gerhard, it doesn't really matter where the symptoms start or how they start. Is that fair to say?
That's fair to say to a certain extent. The magnitude of the expected response, I would not dare to predict precisely. But I would say we have enough evidence that if you damage the neuron some way, this approach should work somehow. That's a little bit more loosely, but that's how we have conviction. And I think nobody who has, studied this field, who would look at our data would disagree with us. So, for example, Lilly invested, among other things, because of CMT and CIPN, just to be clear, because they believe in that too. So that's evidence. It's not just us. I happened to work with another Lilly compound way back then, and I know that they're very interested in CIPN, which is not an indication, it's a side effect of chemotherapy, but it's a big problem. So I'm convinced, and my CMO is convinced, our scientists are convinced, and now it's just the fine-tuning the best response. We cannot yet predict genotype, what CMT type you have, but we have evidence to suggest it should work.
Andy, I saw you take yourself off mute. You have anything you want to add to that?
No, I was going to highlight, Gerhard mentions the CIPN because it seems that this HDAC6 inhibition really has the ability to help it via a number of different ways of the neuron being injured, right? And so that's why there's the confidence that even within the heterogeneity of CMT genotypes, like we're talking about totally different pathologies that HDAC6 inhibition also works on when you think about peripheral nerve damage. So yeah, it's just that I was just giving that context of that's why he's excited about it, because it's totally different diseases and it's working. So yeah, so we're excited about the potential for the broader CMT population.
Oh, that's great.
Yeah.
And so that's a follow-on question that we're getting a lot of those questions. It's who's going to be eligible for your trials? How do you qualify? If I want to be in your trial, how do I find out if I'm eligible, where I can go, what is going to be required of me? We know you've got to start in the healthy volunteers first, and we know you've got a few hoops to jump through before you get into patients. But how are you thinking about the eligibility?
This is an area I want to be careful, right? It's called inclusion-exclusion criteria, as the medical doctor would say, and we're working on this. We have concepts and drafts. We will finalize that realistically by fall, when we have a number of additional meetings with what we call key opinion leaders, very prominent physicians in the field. And we do that in Europe and in the US. I can tell you that Carl and I have already talked about, we want to, of course, also do it in the US. That's important, right? I don't want to talk about politics, but I'd have to say lately it's difficult for us to actually imagine that because, with the uncertainty doesn't help us, which is maybe a topic we comes up later on in terms of what you can also do as patients, because there's a lot of uncertainty. This doesn't help in this field, in any field. But to answer the question, we will have very clear definitions who is eligible, what age range, what severity, we have concepts of that. But we need to test that with a number of really educated physicians in the field, and that's going on.
And some of the insights that we gain from the phase 1 trial will also inform how we think about that phase 2 design. So we will certainly get there.
In your research, have you all found maybe a targeted age or level of nerve damage, level of progression that you might think would be ideal or most receptive to the treatment?
Short answer is no.
Okay.
One thing which struck me when you look at the data, that we actually were able to go in the animals above what they were at 8 weeks, right? So this is an animal which has significant degeneration. We saw the graphs, it's about 60% is left. And you saw that the grip strength is also affected quite. So there is impairment of the animal. And you saw that actually after 4 weeks of treatment. So they're 8 weeks old and then they're 12 weeks old and they get back to above 8 weeks. They don't get all the way back to normal. So that's, if you take animal data and anthropomorphize them, if that's the right word, then you would think this is, suggesting that. But I want to be very careful there, right? Because animals have shorter nerves, right? They're obviously much smaller, so we need to be realistic there. So I don't want to promise anything here. The goal is, to halt the progression, right? And hopefully to also get some capabilities back. But this is early days to make a strong statement there, right? So I mean, I know I got a little bit carried away with the NFL data, but that's actually quite impressive to me, to a point where it's scary, because that could be really phenomenal. If this translates to human only 50%, it would be phenomenal, right? So
Yeah, gives the community something to hope for sure. Ryan, in your perspective, when you look at the entire pipeline of the different drugs that are around this stage of development for CMT, and we just heard Gerhard say halt progression, maybe get some back. What do you think patients should expect? I mean, timing is so hard. We know life is a little complicated right now with timing of clinical trials, but with some of the things going on, how do you think patients with 1A or without 1A should be thinking about maybe the next 5 years or 10 years? Can you do any speculation from the work that you've seen?
Yeah, I mean, it's such an exciting time for the CMT community right now that there's, not just this one, but there's several clinical trials, on the horizon. So, we never know what's going to happen and what's going to get all the way through. And, it's always a longer process than we would hope for. But you have to do all these right steps to make sure it's safe and it's effective, before you can, truly give it to patients. But yeah, I think it's just a very exciting time, and I'm very hopeful that we'll have treatments available soon.
Excellent. And Andy, in your role as a doctor, talk to me a little bit about how you would try and level set expectations for patients for the timeline. And, once we get through phase 1 and then phase 2, like how can you help people understand or have fair expectations, assuming all things go according to plan?
In terms of timing, when we think like an approval is possible,
Or just, how long the phase 2 might take, and then how then that would open up to phase 3, and maybe give a tiny little definition of the difference between phase 1, phase 2, phase 3 approval, except whatever the follow-up is. I forget what that's called off the top of my head.
sure. Yeah. So I'll caveat this by saying, while I did train as a doctor, I've been mostly on the business side of things, but I can answer most of your question.
I was waiting
Yeah.
for you to say that, Andy.
Hey,
I
I
was
mean, yeah, I
waiting
got to
for
be
you.
honest, right? Yeah. Yeah, I'm more about managing my kids' fevers and colds than doing anything more elaborate than that nowadays. But from a trial perspective, so phase 1 is where you're primarily focused on establishing safety of a compound. And as Gerhard mentioned, that's why in this case, we actually start with what in the industry you term healthy volunteers. So these are actually people that do not have the disease of interest that you're trying to target ultimately, but are volunteering to see if there's, just to understand how a person would react to these compounds. So, and then, there's technical terms in terms of like, there's sort of like, we do it in two parts. Like you first start with like a single dose and you go up, and then you move on to the patients getting multiple doses. So that's the phase 1 trial that we are, in the very near term, hope to, be announcing that we've initiated, knock on wood. And, but what Gerhard mentioned was interesting. We will also get insights. He mentioned these biomarkers, that we will also get insights in these early patients around biomarkers of interest that are of direct relevance to the CMT population we ultimately plan to evaluate the potential of our compound in. And so phase 2 is when you really start bringing the patients in, and that's what Gerhard was mentioning. You will have inclusion exclusion criteria for sort of age and a lot of demographic things and severity of disease, or and these are all designed in a way to give the trial, which is very expensive to run, but also it certainly involves patients signing up. It's a global effort, right? So you want to be very smart about how you design those, with the goal of trying to be able to show a difference, or show what we call a signal. But it really means like, can you in the course of the trial show that there's compelling evidence that it's doing something, to keep it simple, right? And then phase 3 is what we call a registrational trial. And so typically, like, if you have strong phase 2 data, it's going to be in a smaller number of patients. And it's really designed to sort of interrogate, can you see impacts in patients? And then the phase 3 is really validating those findings in a larger patient population. Now it's very early to say, because we still have a lot of room to go. But doing, rare disease is a little different, because there are creative ways that you can execute trials in at least in prior years, in conjunction and collaboration with FDA or with EU regulators, to sometimes make those processes not take as long as they might, for example, for a new blood pressure medication, which can take a decade plus. So we still have a lot of work to do, and we still have, we got to start the phase 1, and our goal is to get this to patients as quickly and as responsibly as possible. So hopefully that provides answers. And so any opportunity that we have to collaborate with regulators to do smart things that might shorten that timeline, but in a safe way, we are certainly happy to explore and obligated to explore. So.
Excellent. Thank you. We've had a few more specific questions, but looking that we're almost at the top of the hour, I just want to share my thoughts so kind of summarize what I've heard through these questions. Is a lot of the people in the patient community are hoping that the inclusion criteria includes multiple types of CMT, and it sounds like you're optimistic that it can and will, and that we've got to get through these phase 1 and healthy, just to show the safety, that we're going to do no harm first. Before we can get into patients, but that you all are very driven to do it. And you have some heavy investors behind you, including Lilly, who's not brand new to CMT.
Right.
Ryan, am I correct that Lilly was an investor in DTX and helped accelerate that project forward into Novartis?
Yep, you are correct.
Yeah, so I mean, it's exciting to have them behind this project as well, knowing that they're not new to CMT. So I think, the 84 million is awesome that you've raised it. I think your enthusiasm and care for the CMT community is palpable to me. And as a patient, I'm really grateful for that. And then we just have to keep sitting on our hands and wait for you guys to get through this phase 1. And, we vow as an organization to keep that open communication with our community. And we know that you all as partners will continue to do that with us as well. So let's just wrap up the Q&A session. If I didn't get to your question, and I know there are a lot of them, I didn't want to type and ask questions at the same time. So I'm going to spend the next 5 minutes answering some questions privately, and also give out my information. I'm Susan Rudiger, and you can reach me at Susan@CMTRF. If you have any direct questions for me, feel free to email me. And Laura, I'm going to let you wrap it on up.
Thank you, Susan. So thank you to Gerhard, Andy, Ryan, and Susan for helping CMTRF share this exciting news with our community. And I want to thank all of you for being part of this exciting evening. Your time, your energy, your belief is what continues to drive us forward. The work being done on CMT drug development is driven by your donations and your support. So thank you so much. Together, we're rewriting the future of CMT. Thank you for being part of our community. Have a great night.